I will welcome Oliver and Helen, especially our presenter today, our frog lady, as she likes to go by. She has a lovely background of crispy frog skin, as she likes to say, Cilia, which is pretty cool. Helen is going to be, Helen is an assistant professor at the University of California in San Francisco. She's going to be sharing with us today some research that she's been doing on understanding the role of cilia in DYRK1A, and she's got frogs and human model samples, and depending on the question, she has different models. So depending on what she does, but today it's frogs and humans, and she has presented before in the past in 2021. She's actually her anniversary, 10-year anniversary today. She's been working with DYRK1A. So thank you for doing all the work you do. And we're excited to learn more today. So I am going to let Helen go ahead and start. And just to let everybody know, we are being recorded. So, you know, and if you have any guys have any chats, you know, you have any questions along the way, let us know. I'll look at the chats, and if you, we'll also ask at the end if you have any questions. So we're looking forward to today. Thank you for joining. And of course, I'm Carol O'Neill. This is my first time doing this, so I hope all goes well.
Awesome. Thank you, Carol, so much for introducing me and for all of y'all to have me today. Can you all see my slides? Yes? Yes, good.
Yes.
Yeah, so like Carol said, I've been studying DYRK1A for 10 years now. And what I'm going to try to do today is give you a flavor of the things that we have figured out over the last 10 years, and then also talk about some ideas about where we're going. I love presenting to this group because I actually get some of my best ideas from talking to parents. And so you'll see at one point today, I'll be talking about some stuff we've done with the gastrointestinal tract. And that directly came from one of these roundtables and from parents telling me about their experiences with their families. So please interrupt, ask questions. I'm obviously in a very technical field and sometimes I forget it. So please stop me if things are unclear. I really do want to make this as helpful as possible. I don't take any of that personally. So stop me, interrupt, ask questions. We'll have plenty of time at the end for questions too, if you want to save them up. Okay, great. Yeah, so I'm at UCSF. I study a lot of different genes, actually. So in my laboratory, we study about 100 different genes. We got into this because we were interested in psychiatric disorder genetics. And that was sorry, the WebEx is talking to me. Here we go. That was, these are the lists of all the genes that we study right now. They came from autism genetics, and DYRK1A is one of them. So that's how we started to get into DYRK1A, was through autism genetics. And as you can see, you know, this is about the level of variation in the gene that exists along the population. And so a variant in DYRK1A will increase risk for autism substantially. And so individual variants can be very powerful. And so because I study about 100 genes, that led me to use frogs as a model organism, which is exciting because maybe you don't hear a lot about frogs, but there are some real experimental advantages to working with frogs that motivates why we do it. Unlike fish, you know, you may have heard of zebrafish. They have 2 copies of all of their DNA, so it's a duplicated genome. Whereas in the frogs, they have a much more simple structure to their chromosomes, in which they have 1 copy from mom and 1 copy from dad, just like humans. So they have a diploid genome, more like humans. So that's a big advantage of this system. One mating pair, this is a female and a male, and so one mating pair in an afternoon can lead to 4,000 embryos. So each one of these little dots in this tank is a fertilized embryo. And then this is actually like a 15-centimeter dish. And so each one of these little dots is a fertilized egg that will go on to become a tadpole and then a frog. They have rapid development, so we can watch all the stages of embryo development outside of the mother. And about a week after fertilization, we can get free swimming, behaving, eating, digesting tadpoles that we can use for our experiments. It's very cost effective. My animal costs are orders of magnitude cheaper than a mouse lab, so we can do a lot of experiments and high scale for a lot less money. We can do drug screening, so I'll show you a few examples of that in the context of DYRK1A. Because they're in the water, we can just add any drug we're interested in into the water, and the tadpole will absorb it. And so we can see the effect of that drug on brain development or whatever we're studying at the moment. And then genetics is really great in the frog too. I'll show you some examples later on about how we can make a frog where just the right half of the animal has a mutation in whatever gene we're interested in. So then you can actually compare within the animal what happened when that gene was mutated compared to the other side of the animal that wasn't manipulated. And that's something you can only really do in frogs. So we like that a lot about it. It's also a great model. Like I said, the genetics and the conservation of the genome is really nice. So here what I've done is take the sequence of the protein sequence of the human DYRK1A on the top line, and then put the frog version of the DYRK1A protein on the bottom line. And you'll see that they're very similar. So we're a lot more frog than we like to think we are. And so because these sequences are so similar, we can study the frog version and get some insights into what the human version might be doing as well. So about 10 years ago, one of the first things I did as a postdoc was just say, where is DYRK1A expressed? Like, where is the gene turned on during embryonic development? And then where is it in a cell, right? So I was starting from first principles. And so this is like a, 8-hour-old embryo. This is about a day. It's about a day and a half. This is maybe 4 days. It's maybe 6 days after development. And anywhere that you see this kind of blue purple staining is where the DYRK1A gene has been turned on. And so the only thing you need to take away from this is that it's on in a lot of different places in the early embryo. So this is the head, and this is the tail of what we call a tail bud stage, because it's got a cute little tail bud. But you'll see, the right here is the brain, or the part that's becoming the brain, and that lights up with DYRK1A. The pharyngeal arches are these stripes right here. Those end up becoming the facial bones and with craniofacial structure. There's expression in the developing heart. Strong expression, I'll talk about this a little more later, strong expression in the developing kidney. And then later on, if you look, you can see it. You might be able to see this sort of strong staining right there in the brain. And that's where these neural brain stem cells reside, is right there on that edge. And so you can see strong expression of DYRK1A right in those cells that are actively dividing to become the brain. So that was interesting. It said, okay, there's a wide range of tissues and organ systems in which DYRK1A might be important during development. And then we did pretty standard things in terms of looking within individual cells to say, okay, where is the protein in the cell? And at the time, people really thought about DYRK1A, and still do, and its role inside the nucleus of the cell and regulating what's called splicing, which is how the genes are kind of put together to become proteins. And we did see it in the nucleus, but we also had an interesting new finding that we found it on these structures that protrude out from the cells called cilia. And so I'll explain what cilia are and why we should care about them in a second. But what you're seeing here is these pink hair-like structures are the cilia. And so this is one cell here that's bordered by this green staining, which is also DYRK1A. So this is one cell And out of one cell here on the frog skin, protrudes about 150 to 200 individual cilia that actually coordinatedly beat and generate fluid flow across the skin of the frog. This is very much what happens in the respiratory tract of humans. There's a mucus layer. The cilia beat in order to move the mucus and act as a barrier to pollutants and things that may get into the airways. And so what we found was that when we looked at those cells, DYRK1A was in this really cool dotted pattern all along these cilia that come out from the cell. So that was interesting to us. That had not been described for DYRK1A before we started this work. And so let me tell you why that's interesting and why cilia get me out of bed in the morning. So cilia, almost every cell in the body has at least one cilium. So this is an example. This is actually a human neural stem cell. And you can see this little hair-like structure that comes out from the neural stem cell that's pink right here. That's a neural cilium. It doesn't move like the ones in the respiratory tract. And instead, when it doesn't move, those kinds of cilia are really important for cell signaling. So people often call it the cell's antenna because it's bringing in signals, it's sending signals back to the cell, and really sort of integrating what the environment of the cell and signaling pathways. And it's also very important for how neurons communicate to each other as well. And then other cells in the body are more like the respiratory tract, the fallopian tubes, even sperm tails or motile cilia. And so these are ones that actually actively generate force and actually move mucus and fluid throughout the body. The cells that line the brain ventricles that move cerebrospinal fluid, those are motile cilia that actually circulate the CSF through the brain. So they're very important structures. The cilia field is very much kind of in its infancy, because for a long time people thought that these structures didn't do anything until the 1990s when they realized, oh, they do a lot. And so actually we now know that cilia defects cause a broad spectrum of multi-organ conditions when there are cilia issues in a human. And so you'll see cilia problems will affect many different organs within the body. The ones I'm going to highlight for you today, because they're classic cilia issues that are present in DYRK1A. So a lot of times cilia issues will cause kidney defects, and oftentimes you also see microcephaly in the brain. And then also a lot of gastrointestinal issues can be caused by cilia problems. And I didn't put it here, but also epilepsy. So cilia issues in those neuronal cilia can cause differences in neuronal firing and how the neurons can get excited. And that can lead on its own to epilepsy. So there's a wide range of organ issues that can happen following cilia issues. So I already showed you that DYRK1A is on these motile cilia, like the ones in the respiratory tract. More recently, published this year, we showed that it is also on those primary non-motile cilia in other cell types. This one here is a frog neuron in the gut. And this green is DYRK1A, and the actually yellow here now is the cilium that comes off of that little neuron. And so you can see DYRK1A is present on those primary cilia. And then also we used human cells in culture, which also ciliate. Now the cilia are right here in the white, and you can see that DYRK1A is also, again, right there on the cilium, also in human cells. So it seems to be not just these motile cilia like in the respiratory tract, it also seems to be present on cilia, on neuronal cilia, and many different cell types. that make these single primary cilia as well. So it's there. Does it matter? Is it actually doing something at the cilia, or is it just hanging out? So that's when we go to frogs, and we can make all these manipulations pretty easily. And then within a week, you can have this result. It's the power of frogs. And so this is a control. So this is the normal situation where you have each one of these is a little cell on the skin of the frog, and each one of these cells will put out these cilia that are labeled in pink, and each cell puts out about 150 to 200 cilia, and then you can look up close at just one cell, and you can really see all those cilia. And so that's what it usually looks like. But then using two different ways to perturb DYRK1A. This is a morpholino, and these are CRISPR. The thing that matters is now there's less DYRK1A in both of these situations. And you can see that now these cells that are supposed to have 150 to 200 cilia now only have, a couple. And they're short, and they're not coming out of the cell really, and they're having issues even forming. And so this really told us that DYRK1A is required for the formation of these cilia, and that without DYRK1A, the cilia go away. And this is frog skin. So one of the things we wanted to do then was say, can we actually see something like this in a human that has a variant in DYRK1A itself? Do we see this kind of thing also in humans? So we borrowed from the ciliopathy clinical field here, and took advantage of the fact that in the nose, all of the cells that line the nose have these motile cilia that are moving mucus within the nose. And if those cilia are moving and beating properly, the cells will release nitric oxide gas. Kind of mysterious why they do that. There's some ideas about it, but it's very robust. If those cilia are beating, they produce nitric oxide gas. If there's an issue in the cilia, either the cilia don't form or the cilia don't beat correctly, those cells then produce less nitric oxide gas. And this is so reliable that it's now a diagnostic measure for a ciliopathy called primary ciliary dyskinesia. And there are machines that can measure the amount of nitric oxide gas coming out of the nose, and then give you a metric for how much nitric oxide is coming out of the gas. And then you can compare that to known levels in unaffected individuals, and then also individuals that have been diagnosed with primary ciliary dyskinesia. So yes, I'm a frog lab, but we got an IRB. I'm in a medical center, why not? Then we bought this machine that can actually measure nasal nitric oxide coming out of the nose. And so it's a portable machine. It kind of looks like an iPad, to be honest. And it can measure nitric oxide coming out of the gas in 30 seconds with a non-invasive test. It has this little nasal olive that goes just superficially into the nostril. The individual breathes through their mouth. 30-second reading, and then it will give you the amount of nitric oxide coming out of the nose. I thought that this was particularly attractive in the fact that it was non-invasive, 30 seconds, didn't require a lot from the individual, because obviously with a lot of the folks that I work with, there are intellectual disabilities and behavioral challenges that can make other tests challenging. So of all the kids we've measured now, which has got to be something approaching 100, only one was unable to complete the test. So we did this, James, a wonderful technician in the laboratory, has gone on tour a bit, and people have come to our lab as well. And so here are the first data that came from, and so what we did was we measured the nasal nitric oxide from an unaffected family member, so either a parent or a sibling of somebody with DYRK1A syndrome. And then we also measured, then matched with that person, an individual with a DYRK1A variant. And so we've only done 8 families so far, but that alone is already statistically significant to see a drop in the amount of nasal nitric oxide in these folks. And this red line that I've drawn here is actually the diagnostic cutoff. Anything lower than this would be diagnostic for the ciliopathy primary ciliary dyskinesia. And so to have half of the folks with DYRK1A variant already qualify basically for a diagnosis of primary ciliary dyskinesia really tells us that something is going on with the cilia and that we should be looking at this more seriously for DYRK1A. Okay, so that's kind of basic like cell biology on cilia, but I told you that cilia, then issues can manifest in multi-organ issues. And so that's where the frog really shines.
Yeah.
We can study many different organs in one animal really easily, high throughput, 4,000 embryos a day, and make mutants and see what happens to different organ development within a week. I often tell my students, if this experiment is going to take more than a week, are you really sure you want to do it? Because usually we can finish an experiment within a week and have the result and be able to understand what that gene did when you lose it on various different organs. So these tadpoles, it's a week-old tadpole, they have kidneys, they have a gut, they're digesting food, they have beautiful muscles along their tail, they have a beating heart, they can smell through their olfactory epithelium, eyes, brain, otic vesicles, which help them hear. And so again, today what I'm going to focus on are kind of three major organ issues that are known to have at least a ciliary contribution, or when there is a cilia defect, they often then lead to kidney defects, microcephaly, and GI problems. So with the kidney issues, my only contribution to this paper was this in situ. So this is, again, that image I showed you before, looking at where is DYRK1A on, where's the gene turned on in a developing embryo. It's a frog embryo. That's the head, that's the tail, that's the eye. But when we look at it, what's really interesting is this structure right here, which is the developing kidney. And so we found that DYRK1A was really highly expressed in the developing kidney. And then Rachel Miller's lab at UT Houston went on to then test, it's there, but is it doing anything? And so she manipulated DYRK1A, perturbed it, caused a loss of DYRK1A. And you can see, usually the kidney has this beautiful structure with different kinds of tubules. But then when you lose DYRK1A, now the kidney has serious problems over development. You lose structures, you get kind of cystic structures. And then when she adds back human DYRK1A, that can restore a lot of kidney development, suggesting that the human version can sub in for the frog version in this context. However, if she tries that same thing, but with a patient variant in DYRK1A, it cannot restore kidney development. And so this really tells us that DYRK1A is expressed in the developing kidney and it's important for kidney development. And then Rachel worked with Reza at Baylor to actually then look and see if there are renal or genitourinary tract anomalies in folks with DYRK1A variants. And indeed, once they were looking for it, they could actually find quite a few, these red variants here, quite a few individuals with DYRK1A variants that had congenital issues in either the kidneys or the genitourinary tract. And so that was interesting because a lot of people hadn't directly tried to look for these defects, and without an ultrasound and things like that, often these can be missed if they're not directly looked for. And some of these are things like frequent UTIs, but some of these defects are like agenesis of one of the kidneys, which can be a big deal. So that's one system where this fits with a ciliary mechanism. Cilia are really important for kidney development. Loss of cilia can cause cystic kidneys. And so this is one situation where insights from the frog could really inform and work together with human genetics to actually identify and characterize a new issue going on in the community. Again, so I said kidney defects, microcephaly, and GI. We've done a little work in microcephaly as well. I showed you earlier that DYRK1A was really highly expressed in those cells right along the ventricles of the brain. Those are really the neural stem cells that are rapidly dividing and undergoing cell divisions to be able to make all of the cells of the brain. And so you could imagine if something goes wrong in those cells, that could change the overall number of cells within the brain and ultimately lead to something like microcephaly, where the brain is significantly smaller. And so in frogs, this is our images of the brain and the tadpole stages. I told you earlier that in frogs, we can do this really cute trick where we can make just 1/2 of the embryo lose DYRK1A. And so in this case, this right half of the brain has a lot less DYRK1A than the left half of the brain. So this half of the brain is our control that we can compare to. So this is what the brain usually looks like. And then when DYRK1A is lost, we can see this dramatic loss in the number of cells that are in this region of the brain when DYRK1A is lost. Interestingly, if you overexpress DYRK1A, and of course DYRK1A is in the Down syndrome critical region, so this is more like a Down syndrome situation, now the brain actually gets larger on that side compared to the other side. And then if you put the two together, you can actually restore brain size. And again, the human version can come sub in for the frog version. And now those neural divisions are restored, and you get a brain that is now bilaterally symmetric and has been restored. One of the other things we did was take this situation and then do a drug screen. So like I told you, we just put the drugs in the water, we see what happens. And so we did that with this situation, and we came up with a couple drugs that could rescue or restore brain size when the embryo was treated with that drug. And one of the ones we found, what it was cyclopamine. So it's this drug that inhibits a pathway called sonic hedgehog signaling. We can talk about sonic hedgehog at the end. It has a ridiculous name, I understand. It came from fly work a long time ago. But sonic hedgehog signaling, when I see that, I go, whoa, because the only place in a cell where sonic hedgehog signaling is happening is at the cilium. So sonic hedgehog only happens at the cilium. So to, in an unbiased way, come up with a gene that affects sonic hedgehog signaling, to me says, well, this issue here of this smaller brain, what I haven't shown you is it's actually way too many of those neural stem cells that don't become neurons. They just hang out as neural stem cells. That signaling process of sonic hedgehog signaling that only happens on the cilium is really important for those cells to leave the neural progenitor stage and become neurons. So again, cilia defects often result in microcephaly. Sonic hedgehog only happens at the cilium. So to see that here in an unbiased drug screen really starts to hit home this idea that the cilium might be a very important structure with DYRK1A. So then that third organ system that often presents with issues when there's an underlying cilia defect is the gastrointestinal system. Because in the formation of the gut, neurons have to migrate all the way from, the back of the embryo into the gut in a very particular manner. And that migration of those neurons depends on cilia. So for example, if there's a cilia defect, those neurons cannot migrate. And so it ends up with a gut where the neurons are not even there, which can be very serious. And so actually a conversation with parents at one of these roundtables made me realize just how severe the gastrointestinal issues are in DYRK1A syndrome. So we created a whole project about it, thanks to those parents who shared that information with me. Again, I don't just study DYRK1A, I study, 100 genes, but DYRK1A is here. And so what we did first was just kind of characterize the prevalence. This is a big issue that's going on within these cohorts. And so we used the Simon Searchlight survey data. So thank you to everyone who did those surveys. And when you look at DYRK1A, if you look at the number of caregiver surveys that said that the affected individual had severe GI symptoms. So again, this is like constipation that requires enemas and daily intervention. 20 out of the 30 DYRK1A families reported, yes, very severe GI issues, whereas none of the unaffected direct family members, siblings, parents of these individuals reported similar GI symptoms. So it seems that when there is a DYRK1A variant, there's very often very severe GI issues. And so then we can look in the frog, knock down DYRK1A and say, what happens to the development of the gut? And again, I told you before, if you have a cilia defect, those neurons have trouble migrating. And so in the frog, we can watch those neurons migrate, because again, they develop outside the mother. We can just take a look. And so this is, again, this is the head, that's the tail. Right in here is the developing gut. And everywhere you see these little black spots, those are the neurons that are coming into the gut to innervate the gut so that the gut can move its contents. However, and so now you can see this area where it has migrated into within the gut. However, when we manipulate DYRK1A and cause a loss of DYRK1A, what we actually see is these neurons have trouble making it into the gut, or at least they're delayed in their migration. We did it 3 different ways: one genetically with a CRISPR, one with a morpholino. The morpholinos are always more severe than the CRISPR, but again, here you can say these neurons are really having trouble getting into the gut. And then here we did it chemically with a DYRK1A inhibitor, a kinase inhibitor, TG003. And again, in all 3 situations, what we found was that in the loss of DYRK1A, these neurons have trouble getting into the gut of these frogs, at least. We wanted to take this one step further and say, okay, there's neuron migration defects, but if you look much later, once the embryo has become a tadpole and is feeding and swimming, do you see gut issues there that recapitulate these severe gut issues in humans? So Kate, bless her, she's a graduate student in the lab. She listens to my wild ideas and just tries them. And so I said, why don't you feed them fluorescent beads and watch them move through the gut, and then see how often those beads are excreted by the tadpoles, and maybe we can make that like a scalable drug screen. She did it. She makes my dreams come true. So she fed these tadpoles food with these fluorescent beads. And you look at these mature tadpoles, and here's the green in the gut. So those beads are just swirling around in the gut. And then she created a lot of cool 3D printed tools and stuff to be able to then, in high throughput fashion, place animals in these wells, feed them the beads, put them in these wells, let them secrete and poop out these beads, take the tadpoles out, and then you're left with these wells where you just count the number of beads that came out of the tadpole and start to understand how the gut, so this becomes then a quantitative measure of gut motility. So how well is the gut moving its contents? And that's directly related to the number of beads that end up in the well during this, we now call these porta potties, but how many beads come out within 3 hours? And so she did that. Again, I showed you earlier that DYRK1A drug inhibitor, that's the easiest way to do these. So that's that TG003. This is a control chemical that we add that shouldn't do anything. And so in that control situation, this would be like a typical tadpole, and then each one of these black dots are those fluorescent beads that ended up left behind that had been secreted from the tadpole. And so you get a good number of beads that come out. These are just 3 examples of wells. And so you can see the beads that have been secreted by the animal through the gut. However, when those animals are raised in a DYRK1A inhibitor, so they have a loss of DYRK1A, far fewer beads come out of those animals within that 3-hour pooping time. And so this really then becomes a model for gut motility and to understand and screen for potential drugs that might be able to restore gut motility back to these levels seen in the unmanipulated tadpoles. So the dream would be to, take this and now add different drugs on top of it just during those 3 hours and say, do any drugs now cause the animal to move the contents of their gut? And in my opinion, that might be an interesting therapeutic option to bring back into humans. We had one critical hint during all this. Again, I told you, these cilia are really important for the gut neurons to get there. But these cilia are also really important once the neurons do get there, because in many cases, they are the sole site of serotonin signaling. So a lot of people don't know this, but there's various serotonin receptors. And one of the ones that's really highly expressed in the gut is serotonin receptor 6. And this receptor is known to only localize to primary cilia. And so the gut actually is the main source of serotonin in the body. Serotonin is really important for gut transit and actually moving the contents of the gut. And so all of this kind of told us, okay, DYRK1A is on cilia. There's various serotonin receptors that are only on cilia. If you mess up cilia, you're going to mess up serotonin signaling. So could we potentially find a drug that hits serotonin signaling that might be able to interact with this DYRK1A inhibitor and actually restore gut transit chemically? So Kate took a bunch of different serotonin drugs and just on typical unaffected, like unmanipulated tadpoles, just added these serotonin drugs, did her gut motility assay. And one of them at the one dose we tried, escitalopram oxalate, otherwise known as Lexapro, that one was able to just, in typical tadpoles, move the gut. And so we said, all right, that dose seems to be pretty good at moving the gut of the tadpoles. What if we do that now with the DYRK1A situation? And so this is what I showed you before, though it's a new experiment. But again, you see a lot of these beads come out of the tadpoles in the mock treated, so unaffected, unmanipulated tadpoles. When you inhibit DYRK1A during development, now there's far fewer beads that come out of these tadpoles. But now once you add Lexapro, only during those 3 hours where they're pooping, now you can restore the movement of the gut and actually back to pretty typical levels of gut transit. And so that really showed us that the tadpoles actually are eating. There are beads going into those guts even without DYRK1A. It's just they're having trouble moving the gut. And if you activate serotonin signaling with Lexapro, that can actually move the contents of the gut. Lexapro is a pretty general SSRI. Obviously, I think there are some issues with SSRIs in DYRK1A syndrome. We wanted to go a bit more specific, maybe not all serotonin, but can we maybe, I showed you before that serotonin receptor 6 is very specific to ciliary functions. We said, well, what if we can just manipulate serotonin receptor 6? Can that restore gut motility? Because maybe that could be a more targeted approach to this. So Kate actually found a serotonin receptor 6 very specific activator drug that's called DYRK1A. I don't think it's FDA approved, but we had it, we got it in the lab. And so just like Lexapro, it can help restore gut motility. This was the control. This was inhibiting DYRK1A. This was adding back Lexapro that can now get DYRK1A back to normal levels. And then this was adding that serotonin receptor 6 drug now that also very specifically can add back. And I saw a chat. Yes, these are antidepressants. So Lexapro is like one of the most prescribed antidepressant way. I think maybe not yet. But, the gut is a huge source of serotonin. It's something ridiculous, like 80% of the body's serotonin is in the gut. And it's well known that the serotonin signaling can move the gut. And so these are antidepressant drugs. They're serotonin reuptake inhibitors, things like that. I don't know. So it's very common when somebody starts an SSRI to have pretty severe gastrointestinal problems, But then over the course of a couple weeks, those tend to subside, and then the antidepressant effects kick in. What I think is going on here is actually much more of an acute high dose, quickly in a short amount of time, move the gut and then go on. I think if this were to be developed further for clinical practice, you might really need to deal with dosing and dosing practices so that you don't develop that tolerance that actually you're looking for in an antidepressant situation, where I think here actually it's much more of a acute activation of serotonin signaling in the gut that moves it through. So we're moving this now into rats with DYRK1A issues to try and see if we can now in a very targeted way see if we can take DYRK1A mutant rats, see if we see the same sort of constipation issues, and then if we can add back high dose Lexapro or this serotonin receptor 6 drug, if that can actually restore gut function. But that's in its early days. Okay, so what I've shown you today is that DYRK1A localizes to primary and motile cilia. So it's the cilia of the respiratory tract, but also the cilia of neurons. I've shown you that DYRK1A is required for cilia formation, and that individuals with DYRK1A variants at least have one biomarker of ciliary dysfunction, which is lower nasal nitric oxide. Some of them even at levels that would be diagnostic for primary ciliary dyskinesia, a classic ciliopathy. Cilia defects commonly cause kidney issues, microcephaly, and GI, and we see in the frogs that loss of DYRK1A causes all of these issues, and that manipulating ciliary biology may be therapeutically actionable either through its downstream targets like sonic hedgehog signaling, like we saw in the brain, or through downstream serotonin signaling, like we saw in the gut. Okay, so again, we've been working on this 10 years, but there's many years to come. So what are we thinking about? So some of the outstanding issues, I showed you it's required for cilia formation, but what does it actually do during ciliogenesis that causes those cilia issues? And so Ira Dar's lab in 2021 validated basically everything we did. It was great. And showed that, like, yes, indeed, DYRK1A is required for these cilia to form. And then that lab went one step further in the molecular aspect of it and actually saw that DYRK1A phosphorylates this one protein, CEP97, CP110, that's really important for these, the base of the cilia to form and then go up and make cilia. So there's some insights here that it's active in very early cilia formation through its kinase function outside the nucleus at a target. That's really important for cilia formation. But I think we not forget very early pioneering work from the Jan lab in 2016 that showed, they started in flies and then also showed it in mammalian systems, where DYRK1A, which in flies is named mini brain, is really important for tubulin biology. So these are neurons and dendritic spine kind of things. And when DYRK1A is lost, these get really long. If you have way too much DYRK1A, they get really short. And they went on to figure out that it's because DYRK1A actually localizes along microtubules, which are really important for the formation of these structures. What I haven't told you is tubulin is the main structure of cilia. So just like neuronal structure, cilia are also, the main component is tubulin. And so DYRK1A, they showed, is localized along microtubules, and that actually they think it phosphorylates a serine on tubulin itself, which destabilizes tubulin. And so here that now explains this. When you lose DYRK1A, the tubulin is hyper stabilized, and now you get these long dendritic spines. If you have too much DYRK1A, the tubulin is too destabilized, so you get these really short dendritic spines. And that same process would also affect cilia formation. So if the tubulin is too stable, the cilia cannot form. So I think likely the ciliogenesis problems are both because of this early function in cilia and, what's called basal body biogen. Okay, so the actual base of the cilium being released to come up to form a cilium, but then also pretty basic functions along tubulin, regulating tubulin stability. And so that'll lead to problems with the mitotic spindle, with the cilium, with migration, cilia issue, I mean, microtubule issues will cascade into many different things. So, but there's a lot more to be done here that we're looking into now. One of the things that I think is also a really good use of frogs is what is the therapeutic window? You know, when do you have to intervene in order to restore function? So, you know, can I inhibit DYRK1A all the way to the tadpole stages and then restore DYRK1A function? And then can the gut move? Or do you need to act much earlier to restore DYRK1A function in order to restore organ function? So we're excited to work on that now. We're gonna, and then you know, cilia themselves are very easy to do a drug screen on. And so I showed you earlier that DYRK1A is on the cilium in human cells and culture. That's the easiest place to do a drug screen. So we can actually screen on cilia themselves and knock down DYRK1A, find a cilia phenotype, quantitate it, and then do a quantitative drug screen to try to restore cilia formation. We're also looking into cilia phenotyping in humans. I showed you nasal nitric oxide, but what we really want to do is take nasal brushes where we actually take cells from the nasal epithelium and then look at them and under the microscope to actually look at the ciliary structure and the cilia beating. I've done nothing with seizures. I'm sorry, I know y'all are going to ask me about seizures. I've done nothing with seizures yet, but I want to. So just see if we even can recapitulate seizures in the DYRK1A loss of function frogs, because that also could be something we could drug screen on. And then the other thing that I am gearing up now to work on, both for DYRK1A and a bunch of other genes, is sleep. And so in talking to parents, it'd be interesting to see what y'all's experiences are. But in many other drugs, the kids have real trouble sleeping. And so they're doing melatonin every night, but then the kid is still waking up every night at 3:45 kind of stuff. So I think frogs are a really good model to look at sleep, actually. So we can put the frogs in these 96 well plates. So each one of these is a tadpole. And we can use automated imaging software to actually be able to quantify the activity of the tadpoles. And this blue is the typical situation where at a certain point of development, now they start doing circadian rhythms. And so there's parts of the day where they're very active and there's parts of the day where they are not active. And so it actually cycles through a circadian rhythm. But we've done some preliminary work with DYRK1A showing that they actually become hyper excitable over time and lose some of those rhythms. So I think this is a screenable phenotype that we could, again, try and find some drugs that could manipulate sleep. Okay, so with that, I definitely want to thank everyone in the lab. Yushao Xu, who's now an MD PhD at WashU, started all this work 10 years ago with me in Richard Harlan's lab. And then we carried it on into my own lab, and where Kate McCluskey, a graduate student, has really been focused on it with a lot of help from other folks in the lab. Definitely want to thank our funding sources and our frog-specific resources. So thanks for tuning in. I will be at the Patient Summit in Dallas in July. So I hope to see some of y'all in person there. We will be doing, hopefully, the nasal nitric oxide, expanding that there. So thanks for your attention. Happy to answer any questions.
Okay, well, this is our time for Q&A. I did post that this webinar will be posted on YouTube, our YouTube channel. But if anybody has any questions, if you guys could just unmute yourselves and we'll go ahead and ask some questions. And afterwards, we also have Helen, who wants to maybe present a couple of slides, and I think Oliver has some information as well. So any questions?
Do you have one in the chat? Wasn't a question in the chat?
Karthik, I'll just start. Thanks for the presentation. I mean, it's awesome research. I think it helps for me as a parent when I'm trying to understand what's happening. I think you're talking about microcephaly. That's where, I'm not from the bio field, so it's kind of difficult for me to understand. So I saw that you're saying that you had two sides, and for frogs you can just disturb one side and try it out, right? Maybe if you could spend 1 or 2 minutes to explain that a little bit more, might help to understand it. So.
Yeah, absolutely. So in frog development, right after that first cell division. So you have a fertilized embryo. It does that first cell division. What's really interesting about frogs is that then those cells don't move around during development. So in humans and mice, those cells move around. But in frogs, they stay put. So if we will inject something that will manipulate gene function, so for example, we could inject CRISPR, which will mutate DYRK1A, or morpholino, that'll block protein production, in just one of those two cells, that ends up generating the entire right half of the body. And so then the right half has lost DYRK1A, whereas the left half of the animal still has DYRK1A. So then within one animal, we can directly compare the halves, and then you see the small brain on the side that where DYRK1A is lost. And the microcephaly has been seen in mice and flies, many other model systems before me. It's called mini brain in flies because you mutate in flies and you get a very small brain. And so it's just a way to look at it in frogs now too.
So when you showed that second picture, right? The first picture I noticed it was a smaller brain. Second picture was you put in a human DYRK1A in and it became a bigger picture. I mean, if I'm not wrong, you had a lot of DYRK1A and the brain was bigger or something of that sort, like similar to a Down syndrome. Then what was the third one? That's where like I didn't understand. You had a perturbed one and then you added in more DYRK1A.
Yep. So I was showing them individually, and then I did it together. So I mutated the frog version of DYRK1A, so you lose the frog one, but then on top of that, I added the human one. And now you see together, they kind of cancel each other out, and the human one can sub in for the frog one, and now you restore brain size.
Is that kind of a treatment option in the future or something like that? Like,
so certainly there are a lot of gene therapies that are aimed at doing that kind of things. And so adding back, increasing the levels of DYRK1A that can be expressed. And so there are a lot of gene therapies being developed for other genes and probably also for DYRK1A to actually boost the amount of DYRK1A that are, so absolutely.
Thank you.
And just to kind of add on that, I mean, but what we're saying here is like that happens during development, that is being restored. So, like after the frog is already, a frog, adding back DYRK1A would be kind of a different story. So adding it during development versus, after it's already developed are kind of two different concepts.
Absolutely. So that's why we're thinking about this therapeutic window. When do we have to intervene to actually restore function? And then there was a new paper recently about lithium being able to restore a lot of these function. Lithium is actually one of the few drugs that can actually stabilize cilia. So we're trying to get into lithium too and thinking about, could you add lithium later and restore some of these functions?
We have just a couple more questions in regards to that same thing. Given what you mentioned about GI, is there any current medication that might move stools better than just making them softer?
Gosh, I know. So GI docs will say, give you stool softeners and things like that. I'm not a physician. I don't really know what GI docs will use to move the gut. What we've seen is that these SSRIs do it well in frogs. Certainly psychiatrists know about the issues with SSRIs and sort of inducing diarrhea in their psych patients. But I don't know, maybe.
Okay.
Right. And just to add on that just quickly, I mean, we're talking about different doses. That was high dose from what I think they were looking at in the lab. So I think, you just want to kind of proceed with caution. I think you can certainly ask your treating physician and get recommendations on that. But, just keep in mind that, the doses that are happening, in the frog model, are going to be different than potentially what you're going to see in pediatric or even adult patients.
Mm-hmm.
Yeah, and our mechanism of delivery is totally different. We're putting it in the water and it's diffusing into the gut, right? So it's actually probably like a very high dose, right? So these are just very early days of getting a snapshot of something that maybe we could then actually do stuff with, but it takes a lot more work to get to a point where it could actually be thought about in humans.
And then one other thing was that they asked, how have we ever considered pain thresholds and whether it's ethical? Like
that is interesting to bring up pain thresholds. Cilia are really important for pain sensing. So a lot of times cilia defects will end up with kids that have very high pain tolerances. I don't know if y'all see that in DYRK1A. Yeah, you do. Yeah. So, and especially, I love this one. You need cilia to be able to taste spice. So a lot of times what happens if you have cilia defects, the kids can tolerate really hot, like jalapenos and things that maybe their siblings start like watering and tearing up. So pain, heat, spice, those things are all very much sensed by cilia. Cilia are a sensory organelle. And so, right, have I considered pain thresholds? I've only thought about pain thresholds from that perspective of like, it makes sense that there might be differences in pain sensing because a lot of that goes through cilia, and without cilia you won't feel that kind of pain. Obviously with the frogs and all of our animal work, we have really robust oversight from the university and from the IACUC about what are reasonable amounts of pain for animals to undergo, and we take that very seriously. The stages of development that we're using the frogs at are actually not even considered vertebrate animals because they're so young that they're not thought to really sense pain that much at these stages. So again, a nice use of frogs is that, you don't, those ethical issues are a little bit different, but yes, we think about pain in the animals all the time. I'm a vegetarian, so like I take it very seriously.
we have one more question from Rashid, I think. And Rashid, if you want to unmute.
Yeah, I wanted to thank you. I mean, you say cilia are what get you up in the morning, and I think that for us, this is super engaging information. The way you presented it, I just want to commend you on that. I know I have hundreds of questions, but maybe with time in mind, I'm wondering maybe from both you, Helen, and Oliver, is there ways that we can get more involved if we have an ability to support your work, and even if it's just spreading the word, if there's an opportunity to maybe share how we can learn a little bit more and support more of the work that's really novel.
Thank you.
Yeah, I can chime in real quick. I mean, so, just last year we started our DYRK1A Syndrome International Association grants program. We awarded our first, scientific grant last year, and we are, encouraging researchers to submit grants for us to review to, encourage research and development in the field of DYRK1A syndrome to, ultimately identify a therapy and hopefully multiple therapies that will become available. So yeah, we can definitely keep in touch. I encourage, just supporting, the research program and keeping in touch with the patient group, and we'll continue to support research across all researchers that are interested in DYRK1A syndrome.
Yeah, a lot of the things I focus on come from where I get grants. It's the sad truth of where things are. I have to fund the lab. My lab is completely funded by grants, NIH, and private funding. Private funding right now is more important than ever, as you might imagine. The labs are all very scared right now about what's going to happen with our funding situations. So I think that's where these patient foundations and private sources of funding are really important right now.
Thank you.
Well, Helen, if you don't mind, we have Kellen who has a few slides. If we have a little bit of time here, we can show those slides. I don't know how many you have, Kellen. And just a reminder to upload your genetic reports to RareX, since Oliver wants to get as many reports on the RX as possible. Can everybody see Kellen's slides?
Yeah, hi everyone. I just want to take a couple, hopefully literally 1 to 2 minutes of your time, and just a reminder that we're really trying to collect as much patient information. This is another way to kind of support research, is to, add as much of your because DYRK1A is so rare, every single person matters, and your genetic information and your phenotypic information matters. So we're really trying to push right now to upload genetic reports to RareX, which is the portal I'll show you in a minute. These are the same slides I think I showed last research roundtable, but this is just a reminder. This is your genetic report, and it'll have information on the exact gene mutation in DYRK1A that your family member has. And this really matters because there's all these different types of variants that can all cause DYRK1A syndrome. And researchers need your data to better understand how all these different variants can cause the same disorder and start to tease out whether these variants may cause different symptoms. And also different variants may need to have different therapies developed for them as we get into the therapy development process. So to upload your genetic report, you can just go to DYRK1A.org/participate and click either the login button or the get started button right here. It's under the research tab on the DYRK1A syndrome website. And that will take you to RareX, which is just this patient portal. You create a RareX account, and then you can upload your genetic report, which is the PDF that your doctor gave you at diagnosis. This is a quick video. It's like 20 seconds long. You go into documents, this upload, and then click genetic test, upload, and you can upload the file. It's either just a PDF. I think it'll even accept like a PNG if you take a quick snapshot with your phone. And then you can upload your genetic report that way, and it just pops right up in your RareX portal. And we use this information to study the different genetic variants. And that is it. Here's just a screenshot. Make sure you include all of the different pages. Often these are multiple page reports. And thank you so much. That's all I've got.
Okay, well, is any more questions for our presenter? Thank you so much, Helen. This was very informative. Any other questions? I have one more slide to share. Let me see if I can do that. Hold on one second. And there we go. And if everybody can see that, just remember, you can join our mailer. And again, your participation in our research makes a difference. So thank you all for coming today. Really appreciate it. And hope to see some more of these. These are very, very informative. Thank you, everybody.
Thank you too.
Thank you.
Take care.